Welcome back to our course on the brain. In the last lecture, we talked about how neurons were polarized and that this polarization reflected the fact that neurons are specifically designed to receive information and transmit information in the nervous system. Here, what we want to do in this lecture is to talk about how communication occurs between neurons. We want to understand what the information code is between neurons. Now, a fundamental principle of neuroscience is that neurons are the structural and functional units of the nervous system. This is known as the neuron doctrine. And this neuron doctrine was championed by the most famous neuroanatomist who has ever lived, Santiago Ramon y Cajal. Cajal basically used the Golgi method, which is somewhat ironic, we'll see, to determine that in young animals you could actually follow axons with that specific method to their termination. And from these studies, he established that neurons were structural and functional units. The reason it's ironic that he used Golgi's method is that he and Camillo Golgi, who developed the method, shared the Nobel Prize in 1906. And Golgi believed that the nervous system was not made up of individual cells, but instead was just one connected neural net. And so it was somewhat acrimonious, but they shared the Nobel Prize in 1906 anyway. That neurons are the individual units of the nervous system and separate from other cells was also suggested by Sherrington, who also, of course, won a Nobel Prize. And Sherrington, on the basis of theoretical considerations alone, believed that neurons had to have a space in between them, a gap in between the various neurons in the nervous system, because when he looked at conduction as a physiologist, looked at conduction in nerve trunks, it was much faster than if he looked in gray matter in the brain. So on the basis of that alone, he, in fact, believed that neurons were individual structural units that had to have a space or a gap in between them. Now, this was all the more important since synapses were not actually seen with an electron microscope until the early 1950s. So, we believe that synapses are the places in the brain where one neuron interacts with another neuron. So if we look here at our picture, we can see, in fact, this is one neuron coming down through the brain, crossing, having terminal endings, and this area right here is a synapse where the communication takes place between this neuron and the next neuron in the chain. If we look at one of those terminal endings and look at what a synapse really looks like, this is the axonal ending coming down and wanting to make contact with the next cell, which is here. So this is the axon terminal. These are synaptic vesicles, which are in the end of that axon terminal. And these axon terminals will release a chemical that will interact with the postsynaptic membrane, which is going to be the dendrites or the cell body of that second neuron. So we do know that neurons are, in fact, structural and functional units in the nervous system. So what we want to focus on now is talking about how neurons communicate with each other. If there's a gap between them, then what's responsible for the communication? And a great advance forward in understanding this came about with the discovery that axons actually generate electricity. So this was a very important discovery. So why don't we start our discussion with axons, since it's axons that generate electricity, and that was the first discovery. So let's start there and talk about what happens in axons. So if we look at an axon, there's an electrical signal which gets propagated, and it begins at this place right here, the axon hillock. One of the most important things to remember is that the axon is not just an extension of the cell body. It's a specialized structure, and it's attached to the cell body here at the axon hillock. So an electrical signal is generated here at the axon hillock, and it's going to be propagated down the axon in what we say an all or none fashion. So what this means is that the signal does not degrade, so it's all or none. The all or none signal will travel down the axon in a particular way. It jumps basically from node to node in between the myelin wrappings. Now, if all action potentials of a given neuron are the same amplitude and they don't degrade, so it's all or none, then how is it that intensity would be encoded in the nervous system or encoded in a neuron like this? And what is responsible for the quality of the stimulation? Where we want to learn something about what the code is. Well, the intensity of a stimulus is encoded in neurons by changing the frequency of the firing. So a more intense stimulus causes neurons to increase their firing rate. The quality of a stimulus, on the other hand, is a little bit different. The quality of a stimulus is the result of the stimulation of different kinds of neurons, transmission along specific pathways, and also interpretation by different areas of the brain, especially the cerebral cortex. So let's just think about this in terms of vision. The quality of the stimulus in vision, what we experience ultimately as vision, comes about because an electrical signal is started by cells in the eye that are going to absorb light. So these cells are going to be specialized. These neurons will be specialized to absorb light. Then an electrical signal is going to be transmitted to the brain along visual pathways. That's what it means to say that the brain is made up of these various types of systems. And ultimately, the interpretation, or what mental construct we interpret as seeing, is the result of these pathways eventually reaching the cortex and being interpreted by very specific visual areas of the brain. But the neurons themselves, the neurons, are going to communicate always with these potentials that are generated in axons. So let's go back to this. What is actually happening when this action potential or this electrical signal is being generated in an axon? And to appreciate this and how neurons communicate, we have to understand something about the internal and the external environment of neurons, and how the change in the distribution of particular atoms is going to be the signaling mechanism for transmitting information. So I'm going to use my arms to demonstrate this. And right here, we're going to think of this part of space as being inside of a cell. Remember that neurons are structural and functional units of the nervous system. So they're cells like any other cell of the body. So they have a membrane surrounding them that makes them a separate unit. So this is inside the cell. And neurons, like most all cells in the body, have organelles inside of them. They have special structures that are involved in making proteins, for example, packaging proteins. They also have within them atoms, which are ions, which are basically charged atoms. And that just means an atom which has either gained or lost an electron. So inside of the cell body, you have organelles, and you have ions, and you have other molecules. Now, outside of the neuronal cell membrane, so outside this membrane is what we refer to as extracellular space. So this would be inside, and this would be outside. So outside in this extracellular space, you also have ions. So you could have potassium again, sodium, chloride, calcium, other ions. What is important in understanding how signaling takes place in the nervous system is to realize that the distribution of ions in a cell, in a neuron, is different between the inside and the outside of the cell. So the ions are distributed unequally across the neuronal membrane, so that in what we refer to as the resting state, that the inside of the cell is going to be more negatively charged than the outside. And that is a fundamental principle. So the resting state means the neuron at a time when it is not firing. In its resting state, the ions are distributed unequally across the membrane so that the inside of the cell is more negative than the outside. This charge differential, because remember, ions are basically atoms that have either gained or lost an electron, so they're charged. That means there's a charge differential across the membrane. Now, this differential is maintained in a number of ways. One of the ways it's maintained is that in the membrane itself of neurons are molecules which act as little pumps, and they make sure that the intracellular ion concentration is regulated very carefully. And so that's a very important point. It's very important that the ionic concentrations be regulated because any abnormality in this system or control mechanisms can lead to an abnormal electrical discharge. And an abnormal electrical discharge could set up seizures in the brain or could even kill neurons. So in the resting state, this charge differential is maintained by molecules that actually exist in the membrane that act as little pumps that make sure the right charged ions are distributed across the membrane in a very particular way. In the extracellular space, which is outside of a single neuron, the role in regulating ion concentration is going to be the job of astrocytes. So astrocytes, those little glial cells, those little glue cells that were named star cells, are going to play a critical role in regulating the ionic concentration outside of the neuron. And they do this because they actually act as little sinks for particular charged atoms. And so the extracellular ionic environment is regulated down to the atom by astrocytes. And the intracellular charge is going to be regulated down to the atom by specific molecules across the membrane. So what happens when a neuron is stimulated? Well, when a neuron is stimulated, what's going to happen is that distribution of ions, and you remember that distribution of ions means that there's a differential charge, that this differential in ions is going to be altered across the membrane. So what we have is that if ions now become distributed when the cell is stimulated, when the neuron is stimulated, if this differential charge results in the inside of the neuron becoming more positive, we call that depolarization. If the inside of the cell becomes even more negative than it was before, then it's going to become hyperpolarized. In the resting state, the charge differential is about minus 70 millivolts. So when we depolarize a neuron, it means it moves more positive, and when we hyperpolarize it, it becomes even more negative. When you reach a particular point, and that is when the inside of the cell becomes more positive, to about minus 55 millivolts, you set off an action potential at the axon hillock. And so what we have then is that depolarization, which means making the inside more positive, is excitation in the nervous system. And if you make the inside more negative, so it sends that resting potential more negative, it's called hyperpolarization or inhibition in the nervous system. Action potentials are going to be generated at that axon hillock of the neuron and propagated down the axon in this all or none fashion. It's going to be it's going to be set off here, the action potential being generated here at the axon hillock, and then transferred down the axon, jumping from node to node in between the myelin sheaths. They're very rapid and transient changes in the membrane potential are going to occur at every place where the axon is basically bare here in between these myelin sheaths. Because the action potential jumps from node to node, it's called saltatory conduction. So that means a jumping conduction. Now, the velocity of the propagation down the axon is due to how large those myelin sheaths are around the neuron. The larger the neuron and the greater the myelin, the faster the conduction. If your axon was unmyelinated, the conduction would be very slow. And it's actually very easy to understand why. If you're going to jump from node to node, then the only place you need to change the differential charge is at the spaces in between each node. If you have an unmyelinated axon, then you have to change the charge at each individual point along the axon. So synaptic transmission in myelinated axons is a lot faster than an unmyelinated because it just jumps from node to node. And that's why you can have a neuron and motor cortex, and I can think I want to reach out and touch the brain model, and I can do it almost instantaneously. Yet those action potentials had to go from motor neurons in my cortex down to my spinal cord, synapse in the spinal cord, and go out to cause contraction of the muscle. But it seems almost instantaneous to us because these are myelinated axons, which are very, very fast. Now, this is also going to help us understand why a demyelinating disease, like multiple sclerosis, causes disability in people. This is an autoimmune disease in which autoantibodies are going to attack myelin, and they're going to attack the oligodendrocytes, the cells that make the myelin. So what happens is the axon becomes demyelinated, and that means it can no longer conduct normally. So now, for example, and you can have any system involved, because in the mammalian brain, almost all of the axons are myelinated. There's very few exceptions to that. So what happens in a demyelinating disease is the individual now can no longer control movement, or they're not getting sensory input the way they should because the axons are demyelinated. Now, we used to think that that was the only thing MS was, but we've also learned something else. Axons and their myelin sheath are dependent on each other. So if you have a demyelinating disease like MS, you don't just remove the myelin and change conduction. The axon actually dies, as does the neuron, which is the parent to that. So how is an action potential generated? It's generated at the axon hillock and propagated all or none to the terminal endings. So our next question is, what happens when the action potential reaches the axon terminal endings? That would seem to be the next place to go. So here we have our neuron, and it's propagating an action potential, which there's no decrement in it. It's the same amplitude at every place. And then it comes down here, and in the axon terminals in gray matter, the axon terminals will lose their myelin sheath, and they will form synapses with the next cell. So what we want to know is what happens here at the synapse. So if we look at our synapse again, what we're going to see is our action potential is coming down the axon and then into the axon terminal. And at the axonal ending here, this action potential, which again is electrical charge, causes a particular thing to happen. It causes the presynaptic membrane, the terminal ending, to open up channels that allow calcium influx. So one of our charged ions that's out there in that extracellular space, when calcium comes into the nerve terminal, it sets off a cascade of events which are going to result in the movement of these little vesicles to this membrane termination point, and these membranes fuse with that presynaptic membrane and dump their contents out into the cleft, out into that space that separates one neuron from another neuron. So this neurotransmitter is going to be released there at the axon terminal. So our last question is, what now happens in the postsynaptic structure. So here's our postsynaptic structure, and what we want to understand is the only way this neuron can communicate with that neuron is there has to be some kind of signal that it can read. And what happens? The action potential causes the release ultimately of neurotransmitter or chemicals from this presynaptic terminal. Those chemicals diffuse across the synaptic cleft to interact with specific receptors that are indicated here in this postsynaptic cell. Interaction of the neurotransmitter with those postsynaptic receptors is going to do one of two things. It's either going to directly open channels in that postsynaptic membrane, and that channel is just a protein that controls the flow of ions across its membrane. It's going to do that, so it's going to open a channel, or it's going to bind to another molecule that eventually will also result in different ions being distributed across the postsynaptic membrane. These changes in membrane potential occur in dendrites and spines, so it occurs in the postsynaptic profile. So because of that, we refer to these changes that are taking place at a synapse that take place in the postsynaptic membrane. We call them membrane potentials or postsynaptic membrane potentials. So these potentials are different than action potentials in a number of ways. First of all, they're generated in dendrites and spines. And remember, dendrites are just extensions of the cell body, and spines are just protuberances of that dendritic surface to increase the surface area. Synaptic potentials can be depolarizing or hyperpolarizing. So in other words, they can be excitatory or inhibitory. But last, unlike action potentials, synaptic potentials are graded and decremental. So let's look at what this means. Graded and decremental. Here we have onto our neuron a variety of axonal inputs. So these are axons coming from different places, other nuclei in the brain, synapsing upon this cell right here, either its cell body or its dendrites or spines. Graded and decremental means that the synaptic potentials, which are generated in the dendrites or spines, are graded in that how big they are depends on how much neurotransmitter got released. So they're graded. The action potential was all or none. It had an amplitude that never changed. Synaptic potentials are dependent on the amount of neurotransmitter that's released. That's what it means to say they're graded. Decremental means something interesting. Note that our spines and our dendrites are not myelinated or anything. These are true extensions of the cell body surface. And this means that a synapse way out on here, on this little dendrite, way out here, a little tiny spine, that signal has to change the membrane and ion distribution at each point along the way on its way down here to the cell body and the axon hillock. That means as that signal is transferred, it decrements so that it becomes smaller. Now, what is the consequence of this? What does it mean? Well, it means that it's the sum of all the excitatory and inhibitory input to this neuron that will ultimately determine whether an action potential will be fired at the axon hillock. So you have thousands of synapses coming in and either exciting the postsynaptic membrane or inhibiting the postsynaptic membrane. And then the addition of all these thousands of synapses and their excitation and their inhibition will determine whether an action potential will be generated at the axon hillock. And once that's generated, that action potential will then be propagated in an all or none fashion down here and a synapse here again, postsynaptic potentials, and on and on and on in the chain of neurons. Now, this also helps us understand something else, looking at a picture like this. Helps us understand why it matters so much that particular axonal inputs come into certain domains of the postsynaptic structure. I want you to imagine an axon terminal way out here on the distal dendrites. Well, it's going to have much less effect on firing this neuron than a synapse which is very close to the axon hillock. Because remember, as the synaptic membrane potentials are distributed down these dendrites, they decrement or show a decremental response. So by the time something way out on the end of the dendrite gets its electrical signal down here, it's hardly anything is left. So let's see if you can anticipate something about how the nervous system is organized. If you were an axonal input that wanted to make sure that this cell didn't fire, so under particular conditions, what you wanted to do was make sure that this postsynaptic cell would not fire. Would you want your synapses way out on here on the dendrites, or would you want them right down here as close as possible to the axon hillock? Well, you'd want them right near the axon hillock. And so you would be an inhibitory synapse that would have a major effect that might keep this neuron from firing. So, in development, it turns out, and if this doesn't amaze you, nothing will. In development, different areas of the brain specifically synapse onto different domains of postsynaptic cells where they go. So not only do neurons know where to send their axons, which we talked about before, but actually, when they get to that postsynaptic structure, that nucleus they're projecting to, those axons will synapse on specific domains of that postsynaptic cell. Now, there's a few errors made originally in development, but those errors are going to be taken care of when we have those regressive events that occur shortly after birth. So maybe onto this, if this is a dendrite and these are axons, initially these axons may make 5, that's an underestimation greatly, but some synapses onto this dendrite. Those regressive events may result in only 3 of them remaining. So those regressive events are fine-tuning the connections. You'll lose some neurons that didn't connect with the postsynaptic structure, and also you lose many, many synapses. So we're back to understanding why, when we're born, we have more neurons than we are ever going to have again. And these regressive events that happen after birth are going to fine-tune the system. Some of them will be fine-tuned by experience. Some are just fine-tuned because they're genetically programmed to be fine-tuned. So this is how neurons communicate in the nervous system. And the take-home message here that I think is so important is note, neurons don't do very much. They fire or they don't fire. Or if they're spontaneously active, they either increase their firing or decrease it. That is the information code in the brain. It's basically done by this very simple code. Fire, don't fire, or increase or decrease your firing. Now, if we look at our beautiful image again of a single neuron in the brain, and remember, there are 100 billion neurons in the adult brain. That's after regressive events. You're left with that many neurons. Every single dot here on this neuron represents a synapse coming from somewhere. And it's the addition of all the excitation and inhibition that comes into this neuron. It's integrating all of the excitation, inhibition from all of the other nuclei that would project to just this one neuron that will determine whether this cell will fire an action potential and communicate with the next neuron in the chain. So, to end this lecture, I'd like to paraphrase Shakespeare. To fire or not to fire? That is the question. Thank you.